Welcome to everybody to this second session of the Hudson Virtual Student Open Day. Hopefully some of you have attended the previous ones, but during this webinar, which will go for 30 minutes, this will you'll be muted, but during the question time you can unmute, or you can put any queries that you might have in the chat function, and we'll pick those up either during the presentation or at the end during the question and answers session. And we'll read those out so that everybody knows what the questions are. Now, this is the session for the Centre for Endocrinology and Metabolism, and I'm Peter Fuller. I'm the head of the centre. It sits within a program that hopefully you've seen advertised and will be followed by another Hudson Centre. The Centre for Endocrinology and Metabolism is Australia's leading centre for clinical endocrinology and indeed endocrine research. We're located at Monash Medical Centre, just 10 minutes walk from the university, on the hospital campus. The centre is relatively unique in that it's very closely aligned with and indeed integrated with the endocrinology, the clinical endocrinology unit at Monash Health. Monash Health is Victoria and arguably Australia's largest health service. We have a range of students, both vacation students, honours students and masters and PhD students, and they come from a diverse range of backgrounds, be it science, bioinformatics and indeed medicine. The students come from various departments at Monash University, but also from other universities with close ties with Deakin and also historically with Melbourne University. The centre covers basic discovery research through clinical and translational research into population and health service research around delivery of our outcomes and discoveries. The centre has 6 research groups, and you'll meet each of them today: endocrine hypertension, hormone cancer and therapeutics, steroid receptor biology, sex development, metabolic bone research and clinical andrology. So you can see the breadth of the activity in the centre, and I've listed it, put some asterisks there against each of the centre heads just to show that a number of the centre heads are also active clinicians and head service units within Monash Health. Now, in today's discussion, each of the research group heads will talk about their research. I'll start off, I'll talk about my colleague Rob McLaughlin and my research, and then we'll move through Dr. Jun Yang, and then Dr. Phillip Wong will present the bone research on behalf of Associate Professor Francis Milat, and then Simon Chu and Professor Vincent Harley. We'll talk at the end then about finding a project and supervisor. Brittany, a PhD student, will give you a little of the student perspective of working in the centre, and then we'll take questions, as I said, posted in the chat or potentially by unmuting. Now, the Clinical Andrology Unit is one of the preeminent centres for clinical andrology in the country, headed for many years now by Rob McLaughlin, who's a very distinguished andrologist. And this work addresses issues around sperm production, its regulation, and of course, particularly why sometimes it fails in the context of infertility. There's been a number of studies on the role of testosterone throughout life and the use of testosterone treatment in obesity, type 2 diabetes, increasing problems in the community. Professor McLaughlin's made some significant contributions as part of a larger collaboration around the genetic causes of male infertility, and also is very active in the area of IVF and studies looking at the outcomes of IVF and particularly the consequences for fertility in male offspring of IVF couples. Rob is also the medical director of Healthy Male, which was formerly Andrology Australia. So he has a very active program of outreach, clinical patient education and engagement more broadly. Now, my group is primarily a basic discovery group. I'm interested in adrenal steroids, aldosterone, cortisol. We'll hear a little bit more about aldosterone from Dr. Jun Yang, but these are critical steroids in fluid balance and also the response to stress. They act through nuclear receptors, steroid receptors, which are part of the nuclear receptor superfamily. This is a key part of one of the body signaling pathways, and our area of expertise has been particularly focused on the mineralocorticoid receptor, the receptor for aldosterone. And in that domain, we look at aspects of the structure and the function of the receptor, the relationship of those two things, and how those might be exploited in the development of novel therapeutics. We've also published recently some studies on the evolution of this system and particularly focused on the importance of the mineralocorticoid system, the salt balance required as life emerged from the primordial ooze onto land, and some interesting observations recently there. Obviously, regulation of gene expression is a key aspect of understanding any nuclear receptor, because these are transcription factors. They act by regulating gene expression. So we're interested in mechanisms of signaling and also the role that these receptors might play in tissues other than just those that are involved in sodium transport, such as the breast and breast cancer, and also particularly in cardiac tissues and the cardiovascular system. And to do these studies, we have some novel transgenic mice that contain mutations in the mineralocorticoid receptor, and we have a range of in vitro cellular models, as well as collaborations with structural biologists. We work closely, particularly with Dr. Simon Chu's group, where nuclear receptors also play a fundamental role in granulosa cells, and also the implications of this work flow on to the work of Dr. Jun Yang and the role of aldosterone and mineralocorticoid receptor in hypertension. And I'll pass over now to Jun to talk about her work.
Thank you, Peter, for the introduction. So no surprise, the endocrine hypertension group is interested in hypertension. And it's pretty striking that high blood pressure is the leading risk factor for death, ahead of smoking and diabetes. It's a highly modifiable risk factor, yet 2/3 of patients have poorly controlled blood pressure. Now, what does hypertension have to do with endocrinology? Well, primary aldosterone is a secondary cause of hypertension, and it is an endocrine condition caused by the excessive production of aldosterone from the adrenal glands. And it's significant not just because it's common, but also because people who suffer this condition have greater cardiovascular risk. As mentioned by Peter before, aldosterone and the mineralocorticoid receptor have expression throughout the body, including the cardiovascular system. So diagnosing this condition carefully and treating it with targeted treatment will have significant implications for better patient outcomes. But at present, this condition is rarely diagnosed. Next slide, please, Peter. So the Endocrine Hypertension Service and the research group was set up to integrate the diagnosis and management of primary aldosteronism with research. Our research starts in the community and primary care with outreach to maximize awareness of this condition in the population, to streamlining the diagnostic process within our tertiary care service, to developing cross-disciplinary and multi-center collaborations. We work with neurologists, cardiologists, nephrologists, because they're just as likely to encounter this condition as the endocrine team. We also have multi-center collaborations with large groups overseas in China, America, Germany, Italy, so that our research outcomes are generalizable. We want to learn more about epidemiology of this condition, the health economics of diagnosis and management, the molecular biology of how does aldosterone excess affect cells throughout the body, and how can that help us discover novel biomarkers. Next, please, Peter. So the overall goal of our research program is to diagnose every case of primary aldosteronism in all communities, including the disadvantaged populations, in the most time-efficient and cost-effective manner, and offer targeted treatment for optimal health outcomes. At present, our team is made up of 2 clinician researchers, 2 PhD students, with 2 more clinician PhD students joining next year, and 2 BMS students. We're supported by a clinical research nurse, a senior research assistant, as well as a very busy clinic full of patients with this condition. We also have regular exchange with Associate Professor Morat Young, my PhD supervisor, who heads the cardiovascular endocrinology group at the Baker Institute. So if you have an interest in this area, please do not hesitate to contact me by email. Thank you, Peter. And next, let's hear from Dr. Phillip Wong, who's a senior postdoc in the Metabolic Bone Research Group.
Thank you, Jun, for the introduction. So I'm presenting today on behalf of Associate Professor Francis Millat, who can't be with us today. So the main focus of the group is in bone disease, and we work very closely with Monash Health, which is the largest public health service in Victoria, caring for over 1.7 million patients in Victoria. So whilst we are interested in osteoporosis as a single entity, the research interest of the group is very much focused on the metabolic bone diseases, which means we're interested in the impact of bone disease in patients with chronic diseases across a whole wide spectrum of medical disorders, including chronic neurological disabilities, such as cerebral palsy, transfusion-dependent hemoglobinopathies, such as thalassemia, patients with end-stage renal disease, and those who require transplantation. And also, we have a very strong link with the paediatric bone group at Monash Medical Centre. We often look at patients, young patients with bone disease transitioning through childhood into adulthood. We're also very interested in the role of premature ovarian insufficiency on bone, and also chronic hepatitis and liver disease. So the research, of course, is very clinically focused and translational. All the researchers in the group are endocrinologists and hold consultant positions in the metabolic bone and osteoporosis clinic. We have very strong links with hematology, neurology, renal, and the gastroenterology departments. Next slide, Peter. So this is really a pictorial of what I've just mentioned. So you can see that we're interested in many different sort of diseases across a whole wide spectrum of medical disorders. But I guess the common thread is that these diseases are chronic. The research is multidisciplinary, the research is clinical, and the research is translational. So I might pass it on now to Simon Chu, who's head of the Hormone Cancer Therapeutic Group. Thank you. Oh, thanks.
Thanks, Phillip. Good. So the focus of my laboratory is on a very unique and interesting ovarian cancer known as the ovarian granulosa cell tumours. And these are tumours that, as you can see in the little schematic box there, derive from the granulosa cells that surround the oocyte in an ovarian follicle. And we know the an early mutation event in a very early transcription factor known as FOXL2 gives rise to the majority of the adult form of this disease. And many of these cancers are
caught quite early at stage 1. However, a subset of these tumours will progress to either aggressive or recurrent disease, stage 3, and the molecular events that occur from a stage 1 to a stage 3 disease are relatively unknown. So predominantly that's the focus of my lab, where we're interested in the role of hormones as well as the interaction of various cell-specific signaling pathways in order to understand the development of ovarian granulosa cell tumours, both at the early stage as well as at the late stage. So in my lab, we use various molecular approaches. It's a very molecular focused laboratory. We use molecular approaches, genetic sequencing, bioinformatic analysis to understand the development of granulosa cell tumours in order to find better effective treatment strategies, because what we discover or of those events that lead from stage 1 to stage 3 disease may uncover some of these therapeutic strategies that will be beneficial for patients with this disease. Next slide, please. So I have a couple of projects, little themes that run through the labs that are on offer for honours and PhD students. First one being the molecular pathogenesis of these ovarian granulosa cell tumours. So to establish the genomic landscape of the tumours, to understand what the mutational landscape is like for those tumours, and to explore the role of aberrantly expressed genes in advanced disease. The other project is similarly linked, but whereby there's a common pathogenesis that we've discovered between granulosa cell tumours and another tumour type known as thyroid cancer. And it's the role of this protein known as XIAP, or X-linked inhibitor of apoptosis protein. And so we're exploring the role of this gene in endocrine cancers. And it's a very attractive therapeutic target for novel anti-cancer treatment, because it actually is a very potent anti-apoptotic gene. And so the aim is to identify novel therapeutic targets which, in combination with inhibiting this particular protein, will be utilised in order to investigate whether it's amenable for therapeutic strategies for GCT and thyroid cancer. So the studies involved are in effect precursors that will lead to clinical trials. So you're welcome to contact me if you're interested in any of these tumours. And now we'll move on to the next speaker, Professor Vince Harley from the Sex Development Group.
Thanks, Simon. I work on 3 different areas of sex and gender. The first one is sex determination and diagnosing intersex conditions. So, you know, whether you're a boy or a girl is genetically determined from your sex chromosomes. If you have 2 X chromosomes, your gonads develop into an ovaries and you are female. And if you have an X and a Y chromosome, your gonads develop into testes and you're a male. But sometimes in about 1% of live births, babies are born intersex, and their gonads are typical of neither testes nor ovaries. And I'm interested in what the genetic causes of intersex conditions are. And I'm studying this by actually studying the patient's DNA, so doing human genetics and genomics, to look for the gene differences that are causing the intersex conditions. Developmental biology, by introducing these gene changes into mice, usually, and creating mouse models. And biochemistry and cell biology, where we're trying to get to the mechanisms underlying this. The main gene we've been working on is the gene on the Y chromosome called SRY, which kicks this process off in the week 6 embryo, triggering the development of those gonads down a pathway to testis rather than ovary. The second area I'm working on is a new area that's we're just beginning over the next 5 years. And this is about the fact that many diseases have a sex bias in both prevalence and severity. And I've listed a few that are more common or severe in females and in males. And what we've developed or are developing is a rat model that recapitulates some of these differences, and it's a platform technology that will allow us to investigate any number of diseases where there are rat models to decipher whether the sex bias is a result of sex hormones or a result of sex chromosomes. So this is a very new area. We've made some inroads into this area in relation to Parkinson's disease, where we've discovered that the Y chromosome gene SRY is controlling dopamine in the brain region that degenerates in Parkinson's, and therefore it's controlling movement, and it becomes aberrantly expressed. And we're working on developing a drug that will prevent that cell death in men with Parkinson's in this case. The third area I'm interested in is gender identity. And this is the sex you identify as, whether you identify as a male or as a female. And here we're taking a purely human genetics and statistical approach to examine the DNA from trans individuals, trans men and trans women, in the largest cohort of trans men and trans women in the world. And so we're doing genome-wide associations here to try to establish that the sex you identify at is something you're born with, and the biological basis has a genetic component. So if you're interested in any of these areas of sex and gender, please email me. I'll hand you now back to Peter.
Thanks, Vince. I'd just like to emphasise that if you want to learn more about these projects, you can find those in the 2022 Student Research Project book at the which is online at the hudson.org.au, and the URLs are shown there, and obviously you can get this information also by contacting the supervisor. So when you're looking for a project, identify a supervisor with a project that interests you in the student book, contact the supervisor, introduce yourself. It could be phone, email, Zoom, whatever is currently allowed under lockdown conditions. Check that the project that interests you is still available, and make sure when you talk to the potential supervisor that it's a good fit for you and your background, as well as to meet your expectations. It's important to meet with the supervisor and their team. And if you're excited by the research, then definitely get on and sign up before somebody else does. In terms of signing up to a project for honours and vacation positions, it's a matter of telling the supervisor you want to work on their project, and the supervisor will advise on the next step, including liaising with the relevant university department. Whereas Masters and PhD are somewhat more structured, and you'll find the information online through the Hudson students site shown there, and then there's a series of processes that involve the relevant university, noting that for postgraduate scholarships there are rounds on the 31st of October closing date, and also a second smaller round mid-year. Now, what do we believe you'll gain from research training at the centre? It gives you a qualification that demonstrates your skill in biomedical research. It advances your critical thinking, writing and speaking skills, all of which are part of doing a PhD. It'll give you exposure to and training in the use of state-of-the-art research platforms across a diversity of technologies. And you'll interact with a range of colleagues who are dedicated to improving human health and well-being, which hopefully will be an enriching experience for you. Now, I'd like to hand over to Brittany, who's going to tell you a little bit about herself and what it's like being a student in the centre at Hudson.
Thank you, Peter. So my name's Brittany. I'm a PhD student in my first year, and I'm part of Vince Harley's group with the Sex Development Laboratory. So you may also be hearing that I'm a New Zealand student. So while I'm, for administration purposes, characterised as a domestic student, I've had the unique experience of being a domestic non-Australian navigating my way through academia and research in Melbourne. So I did complete my undergrad in New Zealand and went to Melbourne to complete an honours equivalent course at the same precinct, Monash Medical Centre. Now, part of this course was to conduct research, and I was really lucky to get to do a small research project based in Hudson. And I spoke with the students in this lab, and I heard from these students about the support and collaboration opportunities and the facilities available at Hudson. So I thought that maybe it might be worth my time to also explain to you my experience and for you to hear on a personal note what I've had over the last sort of 6 to 8 months. So while it might sound like we've all got things going on in our individual labs, I've really enjoyed being part of the Centre for Endocrinology and Metabolism because we have a lot to do with one another in our facilities and outside of work as well. So we have cake day on birthdays, which pre-COVID was one of my favourite days of the week. And we'd also have afternoon snacks, drinks, nibbles in our courtyard, pre-COVID lockdown, of course. So we are in a shared open office space, which is a really great experience to be able to lean over and ask questions, like to borrow an antibody or to go and get a coffee. And that seems to be extending to anyone in the centre, researcher or student or lab head alike. So I started in February and I've had a lot going on. So I'm now in the dark pink area here of my timeline. I'm also in New Zealand, having been able to take a couple of days off, but now I've been attending a virtual conference. I'm prepping for conference presentations that I will be doing virtually, and I'm also doing part of our coursework, which is a bioinformatics unit. So you do get the opportunity to also collaborate with other researchers. I'm able to keep in touch, of course, via Zoom, but there is also a really good network of students. So while we have students in our centre, of course, every centre has their own students, which means we also have a student society for Hudson. So we have a variety of networking events, whether they are in person or transitioned to online. So these include things to do with our academic research. So shut up and write sessions where we all log on Zoom and we all do all the writing we need to do. And they are also cocktail and trivia nights. I've been really looking forward to a dumpling night at Ping's Dumplings in Clayton, as well as a student retreat to Phillip Island. So one of my main messages is that our experiences may all be different and they may all end up in different areas, but I agree 100% with what Peter has said about getting in touch with supervisors and contacting them to see what fits you. And another thing though to add to that is to get in touch with the students in the lab to see how they are enjoying their time and their projects and see what advice they can offer you as well, because we have some very valuable information as well about how to get along with your supervisors and how to find the right one. So with that, I will hand back to Peter. Thank you very much.
So the next slide really just emphasises what we've said about contacting research groups and arranging to visit the facilities when that's permissible, of course. So we're pushing our time limit here. We'll move to questions, and I don't know whether we've got any questions at the moment in the chat function or whether people want to, and you can direct those questions to a specific person. I don't know whether any of the others on the line can access the chat function.
No one has asked questions yet, Peter.
Okay, thanks for sharing. So I just perhaps, while we're waiting to see if there are any questions, just put up those contact details again. If there are no questions coming through on the chat or anybody wants to put up their hand or shout out, if you use the hand function, we can unmute you, I believe. If not, we now at 6 o'clock, and I think we're now meant to send you to the next session, which is the Centre for Innate Immunity Infectious Diseases presentation. And thank you for attention, and hope that we'll speak to some of you soon on an individual basis. Thank you for your attention.